Welcome to this in-depth exploration of the Molisch test, a fundamental biochemical test used to detect carbohydrates. Over the next few minutes of this video, we'll break down the test objective, the materials you'll need, the step-by-step process, and most importantly, the positive and negative results that you should look for. The Molisch test was first introduced by a scientist named Hans Molisch, a prominent Austrian botanist and chemist in 1886. Molisch was primarily interested in studying plant substances and their chemical composition, and his work laid the foundation for many important developments in the field of biochemistry. In fact, Molisch test was designed specifically to detect carbohydrates, which, as you may know, are essential organic compounds found in many plants and animals. The test works by reacting the sample with a few simple reagents, most notably a reagent called alpha-naphthol along with sulfuric acid. So Hans Molisch was the one who established this test over a century ago. His work in the late 19th century helped chemists identify and study complex molecules, and the Molisch test remains a vital part of organic chemistry and biochemistry to this day. Although there are more modern methods to analyze carbohydrates now, Molisch tests still serve as a quick and reliable preliminary test for detecting sugars and other carbohydrates in a sample. The Molisch test is primarily used to detect the presence of carbohydrates in the sample. Carbohydrates are one of the four major macromolecules in biology, along with proteins, lipids, and nucleic acid. Carbohydrates include sugars, starches, and fiber. These molecules are crucial in cellular process, energy storage, and structural integrity. In this test, the presence of carbohydrates is confirmed by a reaction that produces a violet color when carbohydrates react with a chemical reagent. The test is based on a chemical reaction between the carbohydrate and Molisch reagent, which is an alcohol solution of A-naphthol. Carbohydrates can be monosaccharides, disaccharides, or polysaccharides, and they can be found in everything from plant fibers to the sugars that we eat daily.
The materials that you will need in order to perform the Molisch test is Molisch reagent, concentrated sulfuric acid, and test sample of 1% solution of the following: fructose as representative of pentose monosaccharides, glucose as representative of hexose monosaccharides, sucrose as representative of disaccharides, starch as representative of polysaccharides, and controlled distilled water as control sample. 5 clean dry test tube holder, test tube stand, and 7 graduated pipette with 1 or 2 ml capacities.
Let's walk through the process step by step. First, we begin by preparing our test sample. You should use about 2 to 3 drops of the sample. For instance, you could be testing a plant extract, a solution of glucose, or even a urine sample to check the presence of sugars. Now, let's add 2 to 3 drops of Molisch reagent to the test tube. It's important to use A-naphthol in ethanol because it's the active compound that will react with the carbohydrates. Swirl the test tube gently to mix the sample with the reagent. Next, we carefully add concentrated sulfuric acid to the test tube. The trick here is to add the acid slowly down the side of the test tube so that it forms a separate layer beneath the sample mixture. This is called the liquid-liquid interface. So why is the sulfuric acid important? Well, it acts as a dehydrating agent. It removes water molecules from the carbohydrates, which leads to the formation of an intermediate compound. The intermediate reacts with the Molisch reagent to produce the purple ring, which indicates the presence of carbohydrates. Now let's look at the results. The reaction is very sensitive and the results can be observed after about 1 to 2 minutes. Although you may need to wait longer in some cases, here's what you should look for. For positive result, if carbohydrates are present in the sample, you will see a purple or violet ring at the interface between the two layers, the layer of sulfuric acid and the aqueous solution above it. This ring forms due to the chemical reaction between the carbohydrates and the Molisch reagent. The purple color is a clear sign that the carbohydrate is present in your sample. For the negative result, on the other hand, if there is no carbohydrate present, you will see no color change at the interface. The two layers, the sulfuric acid and the solution, will remain clearly separated with no purple ring forming. To recap, the Molisch test is a vital tool for detecting the presence of carbohydrates in a sample. The process is simple and involves adding Molisch reagent and sulfuric acid to your sample and observing the results. A positive test results in a purple ring and negative test shows no color change at all. This test is valuable in many fields, from clinical diagnostics to biochemistry, helping scientists and doctors quickly identify the presence of sugars in various substances.